Students, today we will be discussing a past paper. Let me read out the subject code and the component code for you. This is 970042 May June 2023. And from this past paper, today we will be discussing only two questions, question number 1 and question number 2. So let us start. If you see question number 1, it is mainly related to glycolysis, which is the first step in the aerobic respiration. So the main steps of glycolysis you need to recall. We will be talking about them. Meanwhile, you will be able to revisit the topic as well, or you will be able to recall the topic as well. So if you see, the question says glycolysis is a biochemical pathway that occurs in the cytoplasm of cells. In glycolysis, a molecule of glucose is metabolized to two molecules of pyruvate or pyruvic acid, we also can say. The process is outlined in figure 1.1. So if you see, glucose in the first step is been converted into fructose 1,6-bisphosphate, and two ATP molecules have been used, and they have been converted into two ADP molecules. Now, the two phosphate groups from these two ATP molecules have been given to glucose, and mainly this step, we call it phosphorylation. This is the phosphorylation step. Whenever phosphate group is added to some molecule, mainly to make it more reactive, we call it phosphorylation. So addition of the phosphate group is done in the first step. After fructose 1,6-bisphosphate, you can see it has been converted into two molecules of 3-carbon compound that is called triose phosphate. And then there are certain steps which are omitted because as such the discussion was not needed. So each triose phosphate is been converted into pyruvic acid or pyruvate ultimately, and you can see over here that 2 ADP, adenosine diphosphate molecules have been converted into 2 ATP molecules, adenosine triphosphate. It means 2 ATP you are getting over here and 2 ATP you are getting over here. Altogether 4 ATP molecules are produced. At the same time, if you see the coenzyme NAD, so there are two molecules of NAD, nicotine amide adenine dinucleotide, which has been reduced in the form of reduced NAD or reduced coenzyme form. Now, mainly, you have been given an outline, and the question says, explain. Now, I usually tell my students to focus on the command words as well while doing A-level biology. When there is a command word explain, it means you are supposed to explain in a very concise and comprehensive manner depending upon the score which is given. For example, if you see this question is scoring only 1 mark. So for 1 mark, we do not need to write a very big story. We need to be very brief, but at the same time, very clear and to the point, and we should give the most appropriate answer. So let me read out the question. The question says, explain why glucose is phosphorylated at the beginning of glycolysis. If you see, I have written for you the first line as phosphorylation step. What is a phosphorylation step? Phosphorylation step is the step in which the phosphate groups are transferred from ATP molecules to glucose molecule. Now this will not be your answer because this I have written just in order to make a discussion with you. You will have to score 1 mark, and you should be very to the point and be brief and very clear about your answer. So why we are adding phosphate groups? Just to make the glucose active or we are using the phosphate groups for the glucose activation or raising the energy level of the glucose. So I'm underlining this for you. Glucose activation, energy level increasing, and to make it reactive so that it can go through all biochemical pathway of the glycolysis, which is the first main step of the aerobic respiration. So you will be writing any one of them, and in the most appropriate way. It can be used for glucose activation, raising energy level of glucose. It is to make glucose more reactive. At the same time, there is one more option which you can say that it makes the glucose highly unstable so that it can split into two carbon molecules, into two molecules of three carbon compounds, which is triose phosphate. Okay, now the second part, if you see, the command word is suggest. Whenever we have this command word, which is suggest, usually we need to write the reason for the statement, for example. So the question says, suggest one use of the reduced NAD that is produced in glycolysis. So if you see, I already talked about that you are producing two reduced NAD molecules at the end of the glycolysis. At the end of the glycolysis. So please make it sure that you understand like the whole glycolysis process, and you are getting, starting from triose phosphate till the formation of pyruvate, you are getting two NAD, reduced NAD molecules. So you have to talk about suggest one use of the reduced NAD that is produced. What is the use of reduced NAD which has been produced in glycolysis? So the very important point which you people should you should consider or you should keep in mind is that NAD is a coenzyme. It's not an enzyme. And the main role of this enzyme is to get reduced and oxidized again and again. And mainly it is actually carrying the hydrogen atoms or electrons and providing it to the substrate, to the substances and the substrates in the biochemical pathway. Now let's talk about NAD molecule. NAD is a coenzyme, and if you see the one which I have underlined, NAD, and this side also NAD, both of them, they are in oxidized form. After receiving hydrogen from triose phosphate, it becomes reduced NAD and reduced NAD this side as well. So altogether we are getting two reduced NAD molecule. Now, if you see, this oxidation and reduction is very important. Why? Because the triose phosphate is been converted into a different form of molecule, which is pyruvate. So because we had two triose phosphate, so we will be getting two pyruvate at the end of glycolysis. Now let us see what the question says about this. Suggest one use of the reduced NAD that is produced in glycolysis. So reduced NAD is actually carrying, or it's a carrier of hydrogen atoms or electrons, and it is involved in the redox reactions. Of course, if I say redox reactions, it means oxidation reduction reactions. So when the electrons are added or electrons are removed from an atom. So basically it is reducing a pyruvate molecule. So you can just write one use of the reduced NAD depending upon any alternative wording you can use, and will be easier for you to score 1 mark. So you can write carry hydrogen atoms or electrons, or you can say reduced NAD is used in redox reactions, or it is reducing pyruvate. So anyone can be applicable. The last question of this glycolysis question is name. So now let me underline name. So it's not a command word basically, but you just have to say the name. Name the type of phosphorylation reaction by which ATP is made during glycolysis. So if I go up little, scroll down, scroll up. So you can see that initially we use two ATP molecules for starting the biochemical pathway of glycolysis, and our payoff will be 2 ATP. We are getting actually 2 and 2, 4 ATP molecules at the end of glycolysis, but our net payoff will be 2 ATP molecules because 2 ATP molecules already have been utilized at the starting of the biochemical pathway in order to activate the glucose molecule. So name the type of phosphorylation reaction by which ATP is made during glycolysis. So it will be substrate level reactions or it will be substrate linked phosphorylation. You do not need to write the word phosphorylation, or if you write, that's okay. If you do not write, even then it's okay. But what is important, you have to name the type of phosphorylation, and the type of the phosphorylation is substrate level or substrate linked phosphorylation. Now, if you recall the type of the phosphorylation, one is substrate linked and another is chemiosmosis. You must have done in your syllabus. So substrate level phosphorylation means when ATP is produced by conversion of one substrate into another substrate, or by conversion of one substance or a compound into another type of compound, and phosphate groups are actually making ADP into ATP form. So that type of reactions, we call it substrate linked phosphorylation or substrate level phosphorylation. Okay.